Assalamu alaikum. I am going to talk about metagenomics. As we know that there are a lot of microorganisms in environment from which we study only 1 to 5% of microorganisms in lab. But that remaining microorganisms are unable to culture in lab. So metagenomics deal with such unculturable microorganisms. It is also referred as environmental genomics, ecogenomics, or community genomics. Because of metagenomics, we can find diversity of microbes. We can say that metagenomics deals directly with the study of genetic material recovered from environmental sample. This term was used by Joe Handelsman, Robert, and John Clady, and in 1998. There are differences between genomics and metagenomics. Genomics refers to study of genome of single organism, while metagenomics deal with multiple organisms and it is free of DNA. Let's take an example. We take a soil sample. In that soil sample, there are a lot of microorganisms present. We isolate the DNA from them. Then the DNA are cloned in a certain vector. These vectors are inserted into host in which they construct libraries, and then their analysis is being carried out. Three broad type of metagenomic approaches are PCR direct sequence. Normally in sequence reactions, one primer is used, so only one strand is copied. But in case of PCR direct sequence method, two primers are used so that two strands are copied. Whole genome shotgun method. This is used mainly in environmental studies, for example, in seawater sites, abandoned mine sites. It is a technique for determining the entire sequence of chromosome, and this chromosome is based on producing random fragment of DNA that later assembled by computer and different technologies. The last one is a 16S RNA sequence. It is particularly important in case of bacteria that have unusual phenotypic profile, which are also called as rare bacteria, slow bacteria, and uncultivated bacteria. So this 16S RNA sequencing is used for that bacteria, and it is also called as diploid typing, and it is very important from biological point of view. Metagenomics has very wide applications. The first one is the earth science. Scientists are dealing with ecosystem, for example, nitrogen cycle, sulfur cycle, in which certain microorganisms are used. But with the help of metagenomics, we can get the idea of many microorganisms in a particular environment. In life sciences, basically life sciences are dealing with microbiology, ecology, evolution. If we know the function of single microorganism, then through metagenomics we can learn how these microorganisms coordinate with each other, how these microorganisms are expressing their protein, and at what condition they are being expressed. In biomedical sciences, metagenomics helps to identify and study microbial communities to a human body. Basically, it tells us which microbe is causing a disease. As we know that petroleum and petrol products have used at very large scale and they cause pollution. So we use the alternative source of microbiology, bioenergy. Bioenergy provides efficient pathway for various productions. Metagenomics in biotechnology is used to deal with enzymes, genes, and metabolic pathways. And that metabolic pathway leads to drug development. And metagenomics are playing a very great role in the field of biotechnology. In biodefense, metagenomics is used to detect, monitor, and respond bioterrorism, which cause infectious diseases. In agriculture, metagenomics enhance the crop yield, soil health, etc. Before we go to limitation, let's talk about the future prediction. We can also produce new enzymes, new antibiotics, more exotic habitat, which can instantly studied by metagenomics. But there are some limitations of metagenomics, as it does not lead to many procedures. There are genetic tools, a lack of efficient screening method, and inefficient expression of some metagenomic genes. Thank you.